as I cough. I can start recording. Sorry. Anyways, I'm going to introduce myself. I'm Mark Rosenzweig. I'm a patient from 2013. I'm from Detroit, Michigan. And I was, like I said before in many other videos, I have no medical experience. I just learned from others. And I learned from these videos and having doctors from all over the world speak to us. And I also want to go and show you where to find us. Our, so here we go. This is ALK Positive's website. So that's the ALK Positive website, and the place to find us is in ALKtALK. So this is alkpositive.org. And you hit ALKtALK, you scroll down, and you see the join the Zoom room, the calendar, view past ALK Talks, which this takes us to our YouTube page. I highly recommend that you subscribe to this channel, and then this will go back to our page. And then if you hit the calendar, it will take you to all the events that we have. And this is the month of April, but if you click over to the other side, you'll see it'll go to May. So this is where to find us. And also there's so much information here on this website itself. If you go through the home, you can click through all these tabs up top and search everything in the website. So anyways, I want to, first of all, this was a three-year project. Cindy, please mute. There we go. Am I showing? I'm not showing up on your screen, but anyways, this is a three-year project. And I want to thank Dr. Ellie Urban, Jeff Sturm, Dr. Colin Barton, and tonight's presenters, Emily Venanzi, and actually Dr. Emily Venanzi, Professor Mark Muskovich, and Dr. Ken Culver, who always pops in and joins us. So I want to thank everybody, and I'm going to introduce some polling questions. So the first one I'm going to launch here is, what is your type of treatment? All right, I'm going to end the poll. Hey, Summer, can you see me? Because I can't see myself.
I see you, but I have it in a different view.
Okay, sorry.
Yeah,
All right.
Yeah.
Okay, good. All right, so the Lorlatinib is 52%, Lorlatinib is 28%, and that's really about it. So, which is kind of amazing. Brigatinib is only 4%. So you guys can see it right here. And then I'm going to launch another poll, and hold on. Sorry, my computer's acting up. All right, what's the length of your disease? Okay, I'm gonna end the poll. Share the results. So I'm going to lump it together. 44% of us are 2 years or under, and then 3 and 4 years are 26%, and then we got 5 and 6, 14%. So I'm going to stop sharing that, and then go back and launch a couple new polls. If you are taking Lorlatinib, how long has it been since you've been taking Lorlatinib, or also people call it Libreno? So this is only for people taking Libreno. Okay. I'm going to end the poll shortly. Share the results. 50% are 0 to 1. 1 to 2 years is 25%. 2 and 3 years is 17%. And then we go to 5 and 6%. 6 years is 6%. 6 to 7 is 3%. So I'm actually also 6 to 7. So there's 2 of us that are on the call. And then we got a couple of other new polls. What age were you at diagnosis?
And this one we put in because of your feedback. So keep that feedback coming. We really appreciate it. And a few of you suggested that, and that's why we added this. Love it.
Okay, I'm gonna end the poll, share the results. So basically we have 1% 20 to 29, 4% of 30 to 39, 20% of 40 to 49, 50 to 59, 37%, 60 to 69 is 29%, 70 to 79 is 9%. Stop sharing that poll and go back. And this is, I'm going to ask Mark to explain this question. Mark, can you unmute and explain it?
Okay, thanks. Thanks, Mark. So the idea is once you started your TKI therapy, has your disease remained stable? Have you had a partial response where you have had a reduction in tumor volume, but they haven't disappeared? Or have you had a complete response where your tumors disappear completely? Or have you experienced progression since starting your TKI? So those are the four options. Or have you had a mixture of these? That's the fifth option.
Thanks. I feel like I'm a class professor. Sorry, you look goofy sometimes on these ALK talks. All right. Okay. Looks like most of you guys have figured it out. So I'm going to end the poll and share the results. Wow, that's all across the map here. A partial response at 28%. NED, a complete response, 13%. Stable disease response, 29%. Disease progression, 10%. And 29% are a mix of two or more of these responses. So I'm stop sharing. And that is it for our questions and polling questions. And I want to say one thing before I pass over to Summer, and that is these 4 videos so far that we've done, we've had 2,000 YouTube views, number one. And number two is please share them with other ALK patients. Please pass them along to doctors and just tell them about the series. So if a patient gets diagnosed with ALK positive, there's at least a starting point and a progression through the 5 parts of our series, which I think is fantastic. And I'm gonna pass over to Summer Furman.
Hello, welcome everybody. Thank you so much for coming to our last installment of ALK 101. I'm Summer Farman, Mark's sidekick, or ALKtALK coordinator. So again, thank you so much. If you've tuned into the other talks, you know that this has been a long time coming. We've been planning this beginning of ALKtALK. So we're so excited to finally see it all come to fruition. So I was diagnosed in the summer of 2020, stage 4 ALK positive. For those of you that don't know, my father spent his career working in pharmaceuticals and eventually as a biotech consultant in clinical trial development, specifically with lung cancer. Also weird how that happens. But anyway, soon after my diagnosis, we both joined the support group, and my dad did his introduction, as we all do, and Colin Barton quickly scooped him up, or recruited him, and pulled him into the medical committees. So interesting, though, is that even with this years and years of experience that my father had, he confided in me after one of his first meetings that there were a whole lot of very smart patients and caregivers in these medical committees, and he actually felt a little bit out of place. He wasn't sure that he was qualified to sit on the committees. Luckily, he stuck with it. And Emily and Mark are like, yes. His experience in clinical trial development and all of his resources and contacts have been invaluable. But my point is that that was accurate, that there are a lot of very smart people working behind the scenes for all of us. And we have 2 of them with us tonight. So, because we know that ALK does not discriminate, right? No one is immune. It can target any of us. So luckily and unluckily, it has targeted some very smart people, and we have them here. We have Emily Venanzi. She is a PhD with her training in biochemistry and immunology. She was in laboratory research for 10 years. In 2017, unfortunately, she was diagnosed with non-small cell lung cancer, ALK positive. She enrolled in a clinical trial and sits on several medical committees. So we're so grateful for Emily. And then we also have Mark Muscovich, also a PhD, training in biochemistry as well, and genetics, which after watching my daughter go through chemistry, I don't know how you two did it. So it's amazing. Mark has been a professor for 30 years, and he's also worked in pharma and biotech industry for 10 years. In 2020, he was diagnosed with ALK positive lung cancer, and in 2021, he also was scooped up or recruited and joined some medical committees. Together, they work on the Research Acceleration Committee, the Clinical Trials Committee, the Research Review Committee, and a new committee that we have, the Translational Investigation Group. Mark also serves on the ALK Positive Inc. Board of Directors. I have gotten to meet them both in person, and they are absolutely lovely. And with a little arm twisting, they agreed to wrap up our ALK 101 series. So we are so fortunate to have them with us tonight. So thank you both for coming.
All right. Well, Summer and Mark, thank you for the introduction for Emily and me. Emily and I have arranged it so that I'm going to try to begin. So let me share, and here we go. So I'm sure you're all glad that we're at the end. We hope that we provide an appropriate conclusion, because we're here tonight really to discuss the fact that there is a lot of hope out there, actually. There are many, many options, some of which we can access now as patients, even after Lorlatinib, and some of which are further down the line, but we want to summarize those for you tonight. And so the way we want to do that is we just wanted to begin. We've already been introduced. We're going to give just a very brief recap about ALK positive lung cancer. We're going to talk about finding the causes and framing the treatment. Spend more than half of our time about the explicit reasons for hope, the way we are helping ourselves or trying to help ourselves as patients and caregivers, clinical trials that are in progress, and preclinical therapies that are exciting and in development. So to summarize a bit of relevant background, remember, our lungs have to renew themselves, and so there are always a few cells in our lungs that are dividing at a slow rate. But when the ALK tyrosine kinase is turned on in those dividing cells, they start to divide more quickly, and eventually they acquire enough alterations that they become cancerous and out of control. They can spread in our lungs, they can spread to other parts of our body, and those abnormally dividing lung cells form tumors in the lungs initially and then beyond the lungs. But we do have the good fortune that multiple generations of inhibitors of that ALK kinase exist, the TKIs or tyrosine kinase inhibitors. This is a picture. The normal ALK signaling protein is actually bound on the membrane of the cell. That's this little double layer here of the cell membrane, and sends signals by a number of pathways inside the cell. In ALK cancer, what happens is what's called the intracellular or cytoplasmic portion of the kinase, which has the enzyme that actually adds phosphate groups, which is what a kinase does, to target proteins to signal growth. That intracellular domain is freed from the membrane and now begins to signal hyperactively and turn cells cancerous. But our TKIs will inhibit that protein as long as the TKIs can fit into a part of the protein called the active site. And in fact, this is a summary of these multiple generations of TKIs we have. Crizotinib was the first miracle drug for ALK positive cancer, and followed by a number of second generation drugs. Many of us begin now on alectinib, and followed by the third generation lorlatinib. These TKIs will inhibit the ALK kinase, turn down the signaling, turn down the rate of abnormal cell division, and actually promote cell death. of the cancer cells. Those TKIs, though, can be frustrated or blocked from doing their job by changes in the amino acid sequence in the ALK kinase. And so a protein is made up of a chain of amino acids. We have 20 different amino acids in our bodies. And if one of those amino acids in the ALK kinase domain that's expressed in the cytoplasm changes in a way that's problematic for the TKI binding to and inhibiting the kinase, then in fact we get resistance. And so, for instance, if we have resistance to alectinib, we can then move on to lorlatinib. But eventually, most of us will develop resistance even to lorlatinib, and that's why we're talking about life after lorlatinib this evening. So what does this mean? Remember, cancer progression, Colin talked about this, can really be the continuation of cancer growth initially if you continue to progress. It can be the resumption of cancer growth at an existing tumor site, or it can be the beginning of cancer growth at a new site. Those are all progression. The other thing Emily and I wanted to discuss briefly is that when we say life after lorlatinib, we really actually mean life after lorlatinib fails in part, because in fact, even though lorlatinib has failed to control the progressing cancer cells, lorlatinib at that stage is still actually controlling many, many tumor sites in your body. And we know that because many people, if they abruptly stop TKI treatments, will experience what we call a disease flare, where what happens is that the tumors grow rapidly, often at multiple sites, and that can be very problematic for the patient. So in fact, life after lorlatinib usually means life holding on to the lorlatinib treatment and then adding treatments on top of lorlatinib, almost always. This is just a summary to remind us of the types of progression that people experience. There can be progression at a single site or progression at 5 or fewer sites, which is called oligoprogression, or there can be progression at many sites, which we call systemic progression. So, practically speaking, what do we do if we observe progression? So when I experience progression, which I will at some point in the future, by the way, I've had a partial response. So I had a partial reduction in my tumor volumes. I still have some tumors evident on alectinib, and I'm waiting to see how long that response holds. But at some point, I'm sure my cancer is going to progress. And when it progresses, what I'll want to do, if I can, is I'll want to have a rebiopsy of my cancer and a biopsy of new growths. Okay, and when we biopsy, we then withdraw some of the cancerous cells and we analyze those cells and their products using DNA in the microscope or looking at their histology. Progression, in terms of the kind of causes for progression that we experience, sometimes there will be a single change of one amino acid in the ALK kinase that causes the TKI to stop working. Sometimes there will be 2 or more mutations. We call that compound resistance mutations in the ALK kinase domain that stop the TKI from working. In addition, sometimes, as illustrated in this pie chart on the lower right, there can be mutations in alternative pathways that promote signaling that promotes cell division. We call those bypass mutations because they bypass the ALK kinase pathway. Or alternatively, the cell type can change. So our non-small cell lung cancer can change, for instance, to small cell lung cancer. And finally, sometimes, even though we look, we can't see a change in the new cancer growth. Even then, though, your doctor can still recommend alternative approved therapies and or clinical trials that may control your cancer. Practically speaking, then, what do we do when we do this rebiopsy? What kind of rebiopsy can we perform to try to look for the changes that are driving our progressing lorlatinib-resistant cancer? So the idea is to work with your oncologist to see if you can find specific genetic changes. in the progressing cancer cells. And there are three types of biopsy we can perform. One that you may have heard of is called a liquid biopsy. And what that entails is actually taking out a sample of your blood and then sequencing the DNA that's in your blood that has been released by your dying tumor cells. And what we can do then is we can look at the ALK kinase coding sequence, and we can look at the coding sequences of genes in other signaling pathways to see whether mutations in the ALK kinase or an alternative bypass pathways have occurred. We can alternatively, instead of sequencing DNA from blood, we can actually remove, if we can access it, a piece of the new growth itself, a tissue biopsy, where you take a needle and take a core, or you perform a surgery and you take out a chunk of the tumor or the entirety of the tumor, and you can sequence the DNA in the tumor sample. And so you can do a DNA analysis of the ALK coding sequence and the coding sequences for bypass pathways to look again for changes that are driving the Lorlatinib resistant cancer. And finally, if you do have tissue in a core or a tissue biopsy sample, you can actually look under the microscope to see if the cells look different. For instance, a transformation from non-small cell to small cell lung cancer. And if we can see those changes, they usually fall into one of three categories. And these are different categories that you'll want to discuss with your physician when you experience progression. If there are changes in the ALK kinase itself, mutations in the ALK kinase, we call those on-target resistance mutations. They're happening in the target of the TKI, the ALK protein itself. If there are changes that occur in genes other than ALK in these other bypass signaling pathways, we call those off-target resistance mutations, because since ALK is the target and they're in some other gene, they're off-target. They're not in the target of the TKI. They're off-target resistance. And finally, the third term of type of progression we observe is called lineage transformation or cell type change. When your ALK positive cancer cells transform from, say, the non-small cell to the small cell type. And that's sometimes also called a histologic transformation. Histology is looking at the structure of cells and tissues. Those are illustrated here. On the left side, Colin actually used this diagram from this review from Dr. Jamie Schneider. And here you see you can have the ALK kinase with single mutations or compound multiple mutations. We can observe mutations in different signaling pathways that will tell the cancer cells to divide, or we can see a change in the appearance of the cell type and histological transformation. And what this means then is, what do we do? If we have on-target resistance, we are still trying to inhibit the ALK kinase if we can. And so we can look for alternative TKIs, and there are alternatives to Lorlatinib that are approved actually and in trials that we'll be talking about in the next few minutes. If we have off-target resistance, we can use inhibitors of those alternative cell signaling pathways that are stimulating the tumor cells to grow. And if there's lineage transformation, we can try to implement a type of therapy that will kill the new cell type that was not necessarily effective against the ALK positive cancer cells when they were non-small cell cancer cell types. Allows you to frame the treatment of your progression. When you want to pursue the framing of your new treatment, you'll want to talk to your doctor, and you may want to talk to more than just your doctor. And this is where Emily is going to take up the presentation. Emily?
Yes, so anytime that a change in treatment is required is a good time to get a second opinion and consult with someone who is an ALK expert. And these are physicians who spend a lot of their time seeing ALK patients and potentially also do ALK research. And they just have a greater depth of knowledge and experience treating ALK cancer. And they also often have access to clinical trials that might be an option for you. So obtaining a second opinion takes a few steps. It takes some effort. And the first step is to decide which oncologist you'd like to see. And you can learn about what options you have by asking it for advice in the ALK Positive support group, and also looking on the ALK Positive webpage, where on the page with information about the second opinion program, there is a list of about 10 ALK expert oncologists who many of our members have already seen for second opinions. And the second opinion program is a wonderful program that was started by an ALK Positive member and patient and is funded by ALK Positive. And it supports financially people who need some assistance getting a second opinion. So once you've decided who to see, you could contact that oncologist's office to make an appointment. Usually you can find their contact information online. And you'll also need to request your medical records and scans be sent to the second opinion doctor. And that usually involves filling out some forms at your current doctor, signing some papers, and sometimes being persistent to make sure that the records are sent where they need to go. And then finally, when you attend a second opinion appointment, you should come ready with a list of questions to ask. And last week, Colin presented a great list of questions to ask upon progression. And so that's a good place to start. And you should also, if possible, go with a caregiver or another person who can help you remember which questions to ask, take notes, and just help take in all the information that you will get at a second opinion visit. Next slide, Mark. Thank you. Another thing we wanted to talk about before we got into more details about treatment after lorlatinib is what we at ALK Positive and all of you can do to help ourselves and to help other people in the ALK community. So Summer mentioned Mark and I are involved in the ALK Positive Medical Committee, and there are a number of us that are working on different subcommittees to help improve the situation for ALK Positive cancer. The Research Acceleration Committee is working with biotech companies and researchers to try to accelerate and encourage the development of new therapies for ALK cancer. The Clinical Trials Committee works with companies and doctors who are currently running trials or are going to open trials soon that will enroll ALK patients. And we work to help understand better those trials so we can then present them to the patient group, and also to make sure those trials are as patient friendly for our ALK community as possible. The Research Review Committee is the group who analyzes and selects, with the help of ALK expert researchers, which research and clinical trials to fund with all the funds that the ALK Positive members raise each year with all of your fundraising. And finally, a newer group, the Translational Investigation Group, is a group of some of the more science-minded, science-experienced people in the medical committees. And we are starting to work on initiating some of our own research led by ALK Positive to help advance the understanding of our cancer. And now there are also a few initiatives we wanted to point out that you can all get involved in to help move ALK research forward. The first is one which you probably heard about a lot already from Colin, which is the ALK Positive Patient Registry. And this is a survey, an extensive survey that's been put together by ALK Positive in conjunction with the University of Washington. And it's open to all ALK Positive patients, or a caregiver can fill out the survey. And we're attempting to learn all we can about the course of the disease, which treatments work for which people, and also try to learn more about the causes of ALK Positive cancer. So the more people who fill out this survey, the better data set we'll have to learn more about ALK cancer. The second initiative is called the Enigma Plus study, and this is a study from Mass General Hospital and GoTo for lung cancer. And this is a study that you can enroll in remotely. And the Enigma study is looking to collect extra kind of leftover tissue from biopsies that you've had in the past. And that will get sent to Mass General, where they will be able to do some analysis of those cancers and learn more about ALK cancer and other cells within the ALK tumors that way. And then finally, there's the Judith Tam ALK Lung Cancer Initiative at the University of Michigan, which you've heard about on some previous ALK talks. And they're also hoping to learn as much as they can about ALK cancer and improve treatments for ALK cancer by collecting samples from patients, both blood samples and fresh tissue samples when patients are having biopsies. And it's possible to enroll in all of these initiatives now, whether you're having progression or not, and then contribute at the appropriate times. And so we hope that you'll take a look at all three of these and consider participating if you're able. Next slide, Mark. Thank you. So now we're going to switch gears a little bit and talk about the treatments that may be applicable for you once you've experienced progression on Lorlatinib. And we have a long list here, and we've kind of categorized them into three different buckets. So one is treatments that are already approved. Two is clinical trial treatments, and three are the treatments of the future. So things that are still being looked at in the lab, but are not yet available to patients. Go ahead. So these green therapies here are what we're calling approved therapies. So these are probably the most common ones that you will hear about from your oncologist or hear about outpatients being offered once they experience progression. Now, for isolated areas of progression, so Mark said either a single area or oligo progression, a few small areas, those may be able to be treated locally while staying on Lorlatinib. So a small area could be removed by surgery or treated with radiation therapy. On the other hand, if you're experiencing more widespread progression, that often necessitates a change in therapy systemically. And so oftentimes you will stay on Lorlatinib and switch to another therapy, often chemotherapy. Mark mentioned, and I'll talk a little bit more about this, you can sometimes switch to another approved ALK TKI or a different kind of TKI, depending on the mechanism of resistance that your cancer is exhibiting. And finally, we put immune checkpoint inhibitors on here because they are approved therapies for ALK cancer post Lorlatinib. This is the PD-1, PD-L1 checkpoint inhibitors that you may have heard about. There's not a lot of evidence that these are very effective for ALK positive lung cancer. There's still some research that needs to be done on that, but sometimes you will be offered that as an option for therapy post Lorlatinib, either alone or more often with chemotherapy. Okay, go ahead, Mark. So Mark and I mentioned it is sometimes possible to switch to a different TKI from Lorlatinib. And there have been some published reports of ALK cancer patients who've been in this situation. So their ALK protein has mutated in such a way that a different TKI will bind and work to inhibit the ALK function, even when Lorlatinib is not working anymore. So this is again, why we stress the importance of getting a biopsy at resistance, because then you may learn that you have one of these kind of resistance mutations that makes it your cancer sensitive to a different TKI, such as crizotinib or alectinib, or down below gilteritinib, which Mark will talk a little bit more about later. That's a different kind of TKI that's going to be in trial for ALK. Go ahead. So in addition to these approved therapies, clinical trials are also an option after progression on Lorlatinib. And anytime that you're experiencing progression is a good time to investigate clinical trials. And there are several different ways to find out about clinical trial options. One of the best is through your oncologist or a second opinion ALK expert oncologist. We also have on the ALK Positive website a trial spreadsheet that is put together by Ellie Urban, who each month keeps it up to date. And this is an extensive spreadsheet, including trials for ALK lung cancer specifically, also some more general lung cancer trials that ALK patients can enroll in, and those are worldwide. And we also have a few write-ups on the ALK Positive website that were done by Jeff Sturm, highlighting some and kind of explaining some different trials that may be relevant for you. And the Clinical Trials Committee is in the process kind of updating these and looking at revisiting which trials we want to present on the ALK Positive website. And then there's also a more general website called clinicaltrials.gov that is a database listing, all of the clinical trials almost. And it's searchable by cancer type, mutation type. And that website is also very good for finding the contact information for people who are running the trial, either the trial sponsor or an institution where the trial is taking place. So once you've found some trials that look like they might be of interest to you, either your oncologist or you can contact the trial sponsor to ask questions and potentially make an appointment. Next slide, please, Mark. It's important as you're considering clinical trials to really think about whether the particular trial is right for you. So first, each trial has a set of eligibility criteria. So, details about the cancer and your health status. And so you have to make sure that your situation fits with the requirements of the trial. And then it's also a big consideration whether you will be able to participate in the trial based on where it is. So if you're in Boston and you find a trial in California, are you willing and able to do that kind of travel for potentially frequent visits during a clinical trial? This can end up getting somewhat expensive, though there are more and more drug companies that are offering assistance for travel for clinical trials. And there are also some nonprofit organizations that can give some financial assistance for clinical trial travel. Another thing to consider is whether the trial treatment has the potential to be better than your other options, like the more standard options. And that's something to discuss with oncologist or second opinion doctor. And the kind of risk benefit considerations are very personal, and the answer and the decision one person might make about a trial could be different than another, just based on their goals and quality of life considerations. Next slide. So once you've decided to participate in a trial, what does this look like? First, you'll go through an informed consent process where you get pages and pages of information about the trial, potential side effects, the timeline of visits, and you learn all about the trial and then agree to participate by giving your, it's called informed consent. And really you are in control when joining a trial, and you can, if for at any time the trial is not what you expected, or you decide you don't want to participate, you can withdraw your consent and leave the trial. So it's always your choice whether or not to participate in a trial. Once you've given consent, there's a screening process that sometimes involves medical tests just to make sure that you are indeed eligible for the trial. And then most clinical trials require more frequent visits than maybe you would have otherwise, especially at the beginning. So if you're taking a new drug, they want to keep a close eye on you, see how the drug is affecting your health. Clinical trial, experimental drugs in clinical trials are provided by the sponsor of the trial. So you won't have to pay for those or try to get your insurance to pay for them, those are provided for you. In a trial, you'll have more careful monitoring, and that includes often some extra tests. So anything that's above and beyond the standard of care type of monitoring will also be paid for by the clinical trial sponsor. And by participating in a clinical trial, you are really performing a service. You're contributing to medical knowledge of ALK lung cancer, and you're also helping others who have ALK cancer now and in the future. All right, thanks. Next, Mark. All right, so now we'll go back to talking about some clinical trial therapy options, and Mark's going to take over again here.
Right. Thanks, Emily. So I'm going to do a few slides. So the first thing I want to say is these are clinical trials. Every yellow rectangle is a different class of clinical trials. And in every rectangle, there are many times multiple trials going on. So I'd say there's hope on this slide. So there's a lot of hope on this slide because these are all possibilities, many of which are in clinical, all of which are in clinical trials right now. We're going to talk about a few of these this evening. So one that you've heard about before probably, and we're all very excited about, is the Nuvalent NVL-655 new drug that's in clinical trials. Now, Nuvalent is a tyrosine kinase inhibitor, an ALK inhibitor, but it's exciting because it was designed, in fact, to treat versions of ALK that have mutations that confer resistance or that lead to resistance to alectinib and lorlatinib and many other TKIs. And the way we can see that is in this column here that's labeled NVL-655 at the top. You see mostly green squares. Green here means that it takes the least amount of the chemical or a lower amount of the chemical to inhibit the version of the ALK protein that is depicted on the left side here. And so, for instance, you can see that lorlatinib, brigatinib, alectinib are very high concentrations, pardon me, in order to inhibit these versions of ALK that have mutations in glycine 1202. That's what that code means. It's the amino acid glycine at position 1202 in the ALK protein. That's just a shorthand for a mutation, but you can see that Nuvalent is very much more effective than lorlatinib, brigatinib, or alectinib at inhibiting those mutant versions. And so, in fact, this clinical trial is in progress. This is the clinical trial number. In the appropriately formatted version of this file, you can actually click directly on the link, or you can go to clinicaltrials.gov and enter this number in the search field, and you'll find this clinical trial and all of its characteristics. But here is a next generation TKI that was intentionally designed in order to inhibit ALK proteins that are resistant to lorlatinib, alectinib, and brigatinib. And it's now moving into clinical trials, and we'll soon know how the phase 1 and ultimately phase 2 and 3 trials are turning out. A second option that we talked about is gilteritinib. Now, gilteritinib is another kinase inhibitor, tyrosine kinase inhibitors, and in fact, it was identified originally as an inhibitor of a protein called FLT3, but actually it inhibits multiple tyrosine kinases, including ALK. So gilteritinib can inhibit ALK, and here again, the green squares mean that it takes lower concentrations of the compound, the chemical compound, to inhibit a version of the ALK protein designated by the changed amino acid sequences in this leftmost column on the slide. The point here is that for gilteritinib, you can see almost all of these squares are green, including green squares where for lorlatinib and alectinib, we have black and red and yellow squares. Again, if you look at the top, green means that it takes lower amounts of the compound to inhibit a mutated form of the enzyme. Yellow to red to black means it takes more and more of the compound, meaning that it's less effective. And so gilteritinib actually may, again, just like Nuvalent 655, gilteritinib has the potential, to inhibit these mutated forms of ALK that are resistant to alectinib and lorlatinib. and even in some cases, brigatinib and ceritinib. There's a clinical trial being initiated by Dr. Angel Chin at the University of Michigan that should begin enrollment later this year or early next for testing gilteritinib. And so this is another TKI that actually has been approved and is used, for instance, in acute myelogenous leukemia for FLT3 inhibition. Angel is going to see, Dr. Chin is going to see whether we might be able to use it against ALK kinase activation in ALK positive cancers. And then before handing it back to Emily, the last type of clinical trial I want to mention is a new type of drug that's being developed for various cancers. This one is being developed for ALK positive cancer and other TROP2 expressing cancers. It's called an antibody drug conjugate. And so what that means, that cumbersome phrase means, is there's an antibody that binds to a protein that's on the surface of the cancer cell. And what pharmaceutical companies have done is they've taken the antibody and they have linked it physically and chemically to a payload, a small molecule that is toxic to the cancer cell, that will either stop the cancer cell division or will kill the cancer cell. So what happens is the antibody binds to the protein on the surface of the cancer cell. The surface of the cancer cell takes the protein in because cancer cells, all cells, are always removing the proteins from their surfaces because they kind of get worn out, and they pull them into the cell to recycle that protein. And so what happens is when the cell pulls that protein off the surface, it not only pulls the protein, but the antibody and this payload. And when the payload antibody complex gets in the cell, the payload is cleaved off of the antibody, and now it can go and kill or stop development of the cancer cell. So these antibody drug conjugates are really Trojan horses, where what happens is the cellular protein pulls the antibody with the toxin into the cell. The toxin is released in the cell, and it can arrest development or kill the cell. And here again is a clinical trial that is recruiting currently for this TROP2 antibody coupled with an inhibitor of an enzyme called topoisomerase 1, which is required to copy your DNA. And TROP2 is expressed on ALK positive cancer and a number of other solid tumor types. And so this is another type of clinical trial that is in progress. So then let me hand it back to Emily.
Thanks, Mark. So another type of treatment that is also in clinical trials is a different type of immune-based therapy called TIL therapy. TIL stands for tumor infiltrating lymphocytes. And lymphocytes are your immune cells, your white blood cells. And in particular, this therapy uses T cells, which are normally cells that rid your body of pathogens. But in this case, T cells can also recognize and kill cancer cells. So in TIL therapy, T cells are taken from a tumor biopsy. And the hope is that some of these T cells are specific for your tumor cells, but potentially are not working so well to get rid of the tumor because there aren't enough of them, or maybe they're not quite in the right state to eliminate the tumor. So these cells are multiplied outside of your body and sometimes also modified to work better, to be more active and more able to destroy the tumor. And then these multiplied T cells are delivered, infused back into your body, usually after a course of a type of chemotherapy called lymphodepletion chemotherapy, which really just makes space for all of these T cells to be put in. And then the hope is that these T cells will, once they're back in your body, will attack the tumor, continue to multiply, and keep killing any cancer cells that even will pop up later anywhere in your body. And there is a TIL trial right now at Moffitt Cancer Center in Tampa for T cells that have been enhanced with something called CD40 ligand, which enhances their immune response. And this trial is specifically for oncogene-driven lung cancer, so ALK positive and other types of lung cancer with mutations. Next slide, please. Another category of therapies that are in clinical trial now are combination therapies of lorlatinib plus something else, another type of drug to attack the off-target resistance that Mark was talking about earlier. So there are a couple of drugs that are targeting off-target resistance in the MAP kinase pathway, which is kind of signaling pathway that can be activated to overcome ALK TKI to cause TKI resistance. And then there is also a trial combining an inhibitor of MET, which is another factor that can be amplified or turned on in this off-target resistance. And all three of these trials are with drugs in combination with lorlatinib. Next slide, and next trial. Then the clinical trials committee and the medical committee are keeping our eyes on some other clinical trials that are ongoing as well. All of these trials should have some new data presented at the ASCO meeting, which is a big cancer meeting in June of this year. And these are other types of treatment that may be options for ALK cancer post lorlatinib. And so these include oncolytic virus treatment, which is a virus that specifically infects cancer cells and can also stimulate the immune system to kill cancer cells. Also looking at bispecific antibody drugs. This is an antibody that recognizes not just one thing, but two. And in this case, it's binding to two different immune checkpoint proteins. And the hope is that this kind of checkpoint inhibitor interaction or checkpoint binding might be more effective than just the single checkpoint inhibitor that is used now. And then finally, tumor treating fields are delivered with a medical device. So in the case of lung cancer, it's a vest that is worn by the patient, and the vest delivers an electric field that it disrupts cancer cell division by affecting how the DNA divides in the cancer cells. And the idea is that potentially maybe if this looks promising for lung cancer in general, this could one day be combined with TKI treatment. Next slide. Now I'll talk a little bit about therapies that are not quite at clinical trial stage yet. They're preclinical, they're still in the lab, but they're ones that we think are potentially promising for ALK lung cancer. Next slide. And the first one of these is another T-cell based treatment, and it's a little different than the TIL therapy. It's called T-cell receptor engineered T-cells, or TCR T-cell treatment. And Dr. Awad and Chiarle and Dr. Gaynor and Hata are working on this kind of treatment. You heard Dr. Chiarle probably a couple of months ago talk about his ALK positive grant that he received recently to work on TCR-T therapy. And the way this works is that T cells are isolated. Instead of from the tumor, they're taken out of the blood of an ALK cancer patient. And then scientists genetically engineer those T cells to have a receptor on them that specifically recognizes a tumor cell, in this case, an ALK positive tumor cell. And these engineered T cells are then multiplied in the lab. And then, like the TIL therapy, they're infused back into the patient, where the hope is that you'll kind of have an army of T cells then to go and attack your cancer. And these cells are specifically engineered to recognize ALK. And so they would then kill the ALK tumor cells, but ignore everything else and be kind of a very specific treatment. Next slide. Another treatment still at the preclinical stage is cancer vaccines. And you've heard about these from Dr. Chiarle and Dr. Awad in the past. ALK Positive has funded some ALK cancer vaccine research that they're doing. And therapeutic cancer vaccines are different than vaccines for COVID or the flu, in that they're not intended to prevent cancer, at least not at this point, but they're intended to be a treatment for cancer. And there are several different types of cancer vaccines based on how they're delivered. And specifically, Dr. Kiarle and Awad and Dr. Lamb are working on peptide-based vaccines. So these are small pieces of the ALK protein that are injected in hopes of stimulating the immune system to recognize specifically the tumor cells that are ALK positive. So those are in the works, and we hope will come to clinical trial before too long. And then finally, there's been some recent lab data on a new type of checkpoint inhibitor called anti-CD47. And this checkpoint inhibitor, when given to ALK cancer mice, basically, encourages an immune cell in the mice, and we hope in patients as well, called a macrophage, to engulf and destroy tumor cells. And there are researchers, Dr. Hatta and Dr. Weisskopf at Mass General and at MIT, that have shown that combining lorlatinib plus an anti-CD47 antibody can more effectively stop ALK positive tumor cells from growing than lorlatinib treatment alone. So the hope is that eventually this could be a combination treatment that could be especially effective. Lorlatinib plus. So as we've both talked about tonight, there are many reasons for hope. There are doctors, scientists, and patients and advocates really working hard to transform ALK positive cancer into a chronic and hopefully curable disease. We do have some great treatments already, the ALK TKIs, as well as radiotherapy, surgery, and chemotherapy, that are now allowing ALK patients to live many years, in some cases, much longer than they would have, 10 or 15 years ago. So things are changing, and we hope that, and the hope is that the trend will continue and our survival will continue to be even longer. There are universities, biotechs, and pharma companies all working to develop new therapies for ALK cancer and to help us understand our cancer better. The ALK Positive organization is funding some great research and clinical trials, including the gilteritinib trial that Mark talked about before. And we're also working on driving our own research to help get some new questions about ALK cancer answered. And finally, there are, as I presented, multiple therapies being evaluated in clinical trials and also still in the lab in preclinical development. So thank you very much for listening, and we're ready to take some questions.
All right, well, I'm ready to ask you some questions. TPX 0131, what happened to it?
As far as I know, it was found to be fairly toxic to patients, and it was tough for patients to tolerate, so that the company making that drug decided to close those trials.
Okay, if you can't get tissue, a biopsy, and you rely on the blood biopsy, liquid biopsy, how can you determine if you have any amplification of MET?
That's a good question.
So I think that based on sequencing the total DNA that's in the circulation, there's something called copy number variation analysis that you can use computational bioinformatics to look to see if you have more copies of MET per genome equivalent compared to other genes that will only be there in one copy. And so you get more reads of the MET gene than you do of the non-amplified genes. So you can actually make inferences about copy number changes.
So if you can't get the tissue, you think that the foundations of the world, Guardant, other companies could give you that information with blood?
I think that they can generate that information, but as Amy Wen, who's works at Merck and works on biomarker development, would tell us, Guardant, Foundation One, et cetera, can see many things, but they are
Fill in the gap, Emily. What do you say?
they can see many different genes, And they're changes, but they're only allowed by FDA to make a comment on certain genes. And so I don't know specifically if they're allowed to comment on MET amplification, but they will acquire the data for it. So I think it's a matter of whether they're approved by FDA to make that determination. And I don't know the answer.
I have a good question here that I think I have an understanding, but if you're on Lorlatinib and you have brain progression, and that's the only progression, my assumption is you do brain radiation, but I mean, you can't test the brain unless you do a biopsy, correct?
So there are ways to biopsy what's going on in the brain. So you can do a spinal tap and get cerebrospinal fluid. There are endoscopies where they can actually go into the ventricle in your brain and pull fluid from there. And so if DNA is released, you can look at it there. You can actually put a small hole in your skull and do a needle biopsy through the skull. So that there are ways to access the tissue. If you're doing brain radiation, you'd probably want to do stereotactic radiotherapy. So you'd want to avoid whole brain radiation if possible, as Colin mentioned, because of the cognitive deficits and other problems that can arise when you irradiate the entire brain. So you'd want to use gamma knife or some version that gives very localized irradiation only of the metastases themselves, assuming they're of a number and locations that allow that.
Do you think, I can't pronounce it, Lorlatinib will penetrate the brain? Into the brain, would cross over the blood-brain barrier?
Right, I'm not sure. I don't know if Ken's on the call. Emily, do you know about annotation?
I'm on. We don't know, Mark. That's something we're trying to understand better.
Okay, thank you. Hi, Seth. All right. Let me see what else we got. I know we talked to Dr. Awad and Dr. Kilare, and I think they keep on trying to get the trial, but do you have any idea when that might come out?
Emily, do you?
I know they're working on finding a manufacturer for the vaccine at this point. So I think they're hoping to move it along as quickly as possible, but we don't have a definite start date for that yet.
Mm-hmm. But we do think it'll happen in 2023. But
Okay.
As Emily says, it's not fixed yet.
Hey, Ken, since you're on the call, is there the future of ALK Positive Inc. funding research? What does that look like exactly?
Well, I'd say two things, and there are many I could say, Mark. First of all, the ALK Positive organization's done amazing stuff at funding some really important laboratory work that hopefully is going to bring fruit on new ways to inhibit ALK. So we're not just relying on tyrosine kinases, but there are other things. And we've heard some of those on ALKtALK. And second of all, we're expanding what we're going to fund in the clinic already, as was mentioned, gilteritinib and the work up in Boston. But we're really trying to look for really great projects that will expedite the transfer of opportunities from universities to biotechs and from biotechs to FDA approval, so that we're making a difference in the shorter term. And we have projects for the longer term, so that we have a really comprehensive portfolio. That's our idea. I think how that's going to happen in the years to come.
Thanks. There was a great summit video that I watched today that Emily was part of, where Dr. Kamich and Dr. Lovely spoke about the same topic. And what struck me was Dr. Lovely's comment, which is every institution has their own tissue biopsies. And she thought that until we really get a line of one basic location where our tissue is being taken from us, that's really difficult to study all these different variations. As an example, I had a double combination mutation that came out of my neck, and it doesn't show up in any of the combinations that are shown on Nuvalent's chart. So I believe that there's a tremendous amount of combinations. Can someone comment on how many known combinations there might be? Did I stump you guys?
Well, nobody knows. That's for sure.
Yeah, we don't have a number for you, Mark, but it's true. There are many different ways that the ALK gene can mutate during therapy. And there are some that are more common than others. For instance, the G1202R mutation often comes up after alectinib, and fortunately, lorlatinib usually is effective for that mutation. And the Nuvalent drug is working to address some of the most common compound mutations, so where there's more than one mutation. And actually, Nuvalent has in the pipeline another drug to address some of the other compound mutations that arise.
So the people on the medical committees, when do you think that Nuvalent will be giving out some information of how their drug is performing in the phase 1 or trial? Do they ever come out with anything, or is it pretty much they're held to the vest until they come out with a phase 2? Or
They should report some results from phase 1. I don't, Ken, do you have any sense of when that might?
Yeah, I don't know the timing, but if you think about what they told us on ALKtALK before, in their phase 1 trial, which is the first time of being tested in humans, they're enrolling 3 patients, they're waiting. If there's no significant toxicity, then they go up a dose and they enroll 3 more patients. So I'm assuming, since the drug hasn't been showing much toxicity, it's going to take at least most, if not all, this year. I have no personal knowledge of it, but these phase 1 trials are a stepwise process. And I think we will hear more once they reach their dose level that they're going to do for the expansion into the four different ALK positive cohorts. So there's no way to predict, probably even for Nuvalent to know, because it's all driven by what they see in patients as they increase the dose.
Right. What I also say about that is that means that the low doses are probably not showing toxicity or are showing minimal toxicities, which means they can run up to higher doses. And that chart that we looked at a few minutes ago does indicate that that Nuvalent is effective at lower concentrations than many other TKIs. And so I think that's all good. Those are good signals and good signs, but we'll have to wait for the results.
And going back to the 2021 Summit video, I also picked up on TIL therapy that it was going to kind of slowly creep its way into lung cancer, especially ALK, that's going to have to be tested in other types of cancer. Since it's been 22 months since that interview, do you guys think of any updates on TIL therapy?
Well, the trial I mentioned is fairly new, and it's specific for oncogene-driven lung cancer. So in some of the earlier TIL trials, there were maybe 1 or 2 patients that had like EGFR or ALK, and so it's hard to tell really whether that therapy, how effective that therapy will be in patients like us. And so it's good now that there is a trial specifically for oncogene-driven lung cancer, and the TIL technology is advancing. So they're not just taking the cells out, growing them up and putting them back in. They're taking them out, trying to enhance them. Like the trial I mentioned, they're putting in a kind of an immune enhancer into the T cells to try to make them work better before they put them back in. So it's the next generation of TIL therapy is now in trial.
Okay.
Right.
That's good
And
news.
then we actually also happened upon a paper recently on the Research Acceleration Committee. It is possible to isolate from blood, from whole blood, cells that are going to be likely to interact with your tumor. You can actually mix magnetic beads with your blood and then pull out the cells that stick to the beads. And you can actually make those beads so they will help you pull out cells that are likely to attack your tumors. And so it means that without having to go and cut out a piece of the tumor, which can be very hard for some. I have, for instance, my primary site is regarded as inoperable in my lungs. You could still have the option of isolating these TIL cells from the circulating blood and not having to go to a chunk of the tumor itself. So that's very early days, but it's very promising.
ADC, is there different companies that are making ADC, and what can you tell us about maybe any of the outcomes or any of the advantages or disadvantages between the companies?
Right. So, I mean, so the basic idea is that the antibody drug conjugate has selectivity based on the antibody and which fraction or which types of cells in our bodies express that protein the antibodies against, right? And so, for instance, one of the things that the Translational Investigation Group wants to do is we want to actually create parts lists for ALK positive tumor cells. And we can do that by a technology called RNA sequencing, where we sequence all the messenger RNAs in the cells, we interpret them in a computer, and then we compare those to the proteins known to be on the surfaces of human cells. And we can actually predict, hypothetically, all the cells that are in ALK positive tumor cells, all the proteins that are on the surface. And so we could try to make antibodies against those proteins. So the antibody of an antibody drug conjugate can provide selectivity. If we can find something only in our cancer cells, it's very selective. And then what happens is, depending upon the toxic payload you've coupled to the antibody, you can have different effectiveness in killing the cells. The challenge is right now, people are trying to figure out
The doses, I'm
the
assuming.
antibody. Because
You froze for a second. You
Oh.
said they're trying to figure out what?
they're trying to figure out how much of the toxic chemical to attach to the antibody. And what you want to do is you want to minimize that to maximize cancer cell killing and minimize toxicity to other cells, right? And so companies are having a hard time figuring that out, but they're working on it intently.
Gotcha.
Mhm.
This
Hey,
question.
Mark.
Yeah.
It's
I
Ken.
was gonna,
Can I
I was
co-
gonna ask you a question, actually. Go ahead.
I want to come back to the chart with the green, red, and yellow and black squares on it. I want to make sure that the community isn't over-interpreting that. Not every ALK mutation, combination mutations is on that list because it'd be way too long to get on a slide. Second of all, we know that within an ALK patient's tumor, more things are changing than just mutations in ALK. So this isn't like a recipe where you find out your biopsy result, you run your finger down the column and you say, aha, this is what you're going to get next. That's not in the treatment guidelines. So it's a guide to physicians and patients, but it isn't a recipe for exactly how someone should be treated.
This question, Ken, goes to you, and that is, you're newer to the scene, and with your experience that you have in your background, did you ever think that you would be meeting these people in these groups in the medical committees? I mean, are you blown away with the when you came into the group and you started learning the processes that we've been doing?
Well, it's a very impressive organization, ALK Positive, and I've been speaking with other lung cancer groups. Saturday, I was on a whole seminar that Cure put on for lung cancer in general. And what I'm learning is ALK Positive as an organization is a leader in patient-driven research, education, working together for the common good. It's very impressive, Mark. I had no idea that, and I think ALK Positive is setting the standard for exactly what a lung cancer patient-driven group should be. So I'm so thrilled to be a part of it as a witness and hopefully a contributor.
Great. Well, hats off to everyone who's been involved in these series, and I really, really think that this is kind of like a learning stepping stone that we're doing in our ALK Talks. We've had, I believe this is our 43rd ALK Talk. We've had 22,000 people on YouTube. We've had over 6,000 people attend. And between that and our monthly calendars of events and all the medical committees, I'm proud to say that I really think that this group is amazing. So thank you so much. And I want to thank everyone for coming. Ken, Emily, Mark, Summer, and everyone else. Thank you so much.
Wait, we got to give a sneak peek.
Yeah, I think that Judith Tam is also in the audience. I want to thank Judith for her work with the University of Michigan. And the next ALK Talk is on the 7th of May in summer. What is that?
That would be with Triage Cancer. It's actually our first sponsored ALK Talk, and Pfizer is sponsoring it. It'll dig into cancer finances and talk about obtaining Social Security disability, using your own disability insurance if you have that, and then eventually acquiring Medicare. So definitely, I know I have a lot of questions myself at this point. So I've been testing the waters with them, and they're a great organization. So that should be a really interesting talk. Then we'll have a break for a little bit, a couple weeks after that, because we've been hitting you hard. But yeah, that's next Sunday.
Thanks, everyone. You can unmute and say hello to everyone.
Thank you, Emily. Thank you, Mark. Thank you, Mark.
Thank
too.
you, everybody.
Thank you, everybody.
Thank you guys so much for
Thanks,
that.
everybody.
Thank you guys.
Thank you so much.
Thank you.
Thank you so
Thank
much.
you.
Take care, all. Thanks.
Bye.
Thank you, Ken.
Thank you.
Bye, everyone.